I think it's live now.
Good stuff.
Give this. I mean, I have it on my phone right here just to see if it's live on YouTube in the account. And it says it's live. Yeah, perfect. All right, I guess it is live.
Nice.
Well, welcome everybody. Playing Games Podcast, episode 14, and today we have a very special guest, an actual researcher on the topic of Sarcomerogenesis. Thank you, Avery, for joining us. How are you doing today?
Pretty good. Yeah, a little tired, but not too bad. Yeah.
Day at the lab or anything?
Yeah, typical day. So all good. Yeah.
I'm personally trying to get my hands on into a lab. I'm still undergrad, so.
Gotcha. Good stuff. Yeah.
But yeah, well, before we get started, why don't you tell us a little bit about your background and kind of what led you to the topic of sarcomerogenesis specifically, because I think that's what you focus on.
Yeah, for sure. Yeah, so I started in undergrad in a program called Human Kinetics at the University of Guelph, which is basically a kinesiology program. It's just called Human Kinetics at Guelph. So yeah, I did that for I did like regular 4-year degree, but in my 3rd year, I joined Dr. Jeff Power's lab as an undergrad. And I did a project where we were looking at the effects of isometric training at a short versus a long muscle length on adaptations in muscle architecture and muscle function. So the main thing we were looking at is if we could like cause fascicle length to get shorter by isometric training at a short length versus longer by isometric training at a long length. And we use ultrasound to do those measurements. So we were sort of inferring like adaptations in serial sarcomere numbers, though, like addition versus subtraction of sarcomeres in series. And yeah, the like foundational research for that was like research on sarcomerogenesis from like the 70s and 80s. So when I moved into my master's, still in Jeff's lab, I transitioned to working with animals, where we could really look into the like that topic, like looking at the actual sarcomere number to see if we could observe increases in sarcomere number, so sarcomerogenesis with different interventions in rats. So that's what I've been working on since then. Kind of just moved right into my PhD out of that. And yeah, think I've been doing some decently cool stuff since then. Right now I'm working with actually old rats, so I'm looking at if sarcomerogenesis is as possible in old age as it is in like younger individuals. So yeah, that's where I'm at
Cool.
now.
Yeah, I've read some of your papers. Great work. Really
Yeah.
interesting thing. Topic I'm personally passionate about. So, well, besides that, I mean, I guess we could get started with a very simple foundation on what is sarcomerogenesis. I think I
Yeah,
pronounced
for
that right.
sure.
In a simple context, but then a more complex definition, because I think people have heard like a very simple definition over social media. So,
Sure,
I
yeah.
mean, if you want to give it a shot.
Yeah, so I guess, yeah, the most simple definition is addition of sarcomeres in series. But yeah, what that means is it comes about by, let's say, it's usually interventions that are stretching the muscle or putting it under some kind of tension that are causing sarcomeres to be overstretched and past their optimal length for like actin and myosin overlap. And that imposes a stress on the muscle that induces all these signals that physiologically stimulate it to add sarcomeres in series along its length so that it will, reduce the tension that's developed by the sarcomere, because now each sarcomere along the whole muscle length will be at a shorter length than it was when it was previously stretched out. And also provide better actin-myosin overlap. So ultimately better force production now in that overstretched length. So the muscle has responded to this stretch or increased tension stimulus by adding sarcomeres along its length to sort of improve its function at that more stretched length. I guess, yeah, that would be the more complex definition. Yeah.
And I mean, I hope for anyone watching this, they're familiar with what a sarcomere is.
Yes, that's true. Yeah. So yeah, I guess.
I mean,
Yeah, I guess I can provide a more. Yeah, that's true for anyone watching. So, yeah, the sarcomere is basically the basic building block of your muscle. So it's all your muscle strength at like the whole muscle level is developed in total by thousands or hundreds of thousands of sarcomeres, even that are all in your muscle working together, producing that active tension that allows you to lift things up or run or jump. That's where the force comes from, just from all those little building blocks that are the sarcomere.
yeah, it's pretty incredible how those little things they like
Yeah.
produce like what piconewtons of force, and then here we are bench pressing hundreds of
Exactly.
pounds for something.
Yeah.
It's pretty impressive.
Yeah.
So then this leads me to the following question. Now that we know what sarcomerogenesis is, this sort of adaptation that happens in muscle, what type of task or stimulus causes this sort of adaptation in muscles? And what are the functional benefits that the muscle can gain from this adaptation?
Yeah. Yeah. So the main one that we look at that would be the most applicable to like a training context is eccentric training. So any resistance training program that would emphasize the eccentric phase of the lift. So that would be when you've already, like, I'll use bicep curls as an example, just because it's visual. You've lifted the weight up, that's your concentric contraction, but now you're going to be lowering it back down, sort of working with gravity. That's the eccentric phase. So resistance training programs that emphasize that part of the contraction, because you're stretching the muscle while it's loaded, that provides that big stretch stimulus to add sarcomeres in series. And there are a few other examples too. So like I talked about, isometric training at a long muscle length could also potentially, there's no direct evidence that it does, because we would need to do some animal studies to confirm it. But if we look at ultrasound measurements where we're just looking at fascicle length, which could be an estimate of serial sarcomere number at a larger scale, those would suggest that, yeah, isometric training, again, at a long length, because your muscle is stretched out and then you're generating the tension, that could also potentially add sarcomeres in series. But yeah, the big one we look at is eccentric training, eccentric exercise. So,
Okay, and
benefit, yeah, that was the second part of your question. So, yeah, so main functional benefits would be that because you're adding more sarcomeres along the length of your muscle, you're basically force length relationship. So the force length relationship is how much force your muscle can produce at a given muscle length. So there's an optimal muscle length because of optimal actin-myosin overlap in an individual sarcomere. So when you add a whole bunch of sarcomeres along the length of the muscle, you're basically widening the range over which your muscle can be closer to that optimal force production. And along with that, that could make it so that your muscle can produce more force throughout the range of motion. So like during an active, like shortening contraction. So that could also potentially improve the power that your muscle can generate during movement. So those are like the main functional benefits that we think about.
right, and I mean well, sarcomeres can be added in series. They can also be added in parallel, right?
That's right. Yeah, exactly. Yeah,
Which we'll get into later on.
yeah.
But I guess the difference would be that when the sarcomeres are added in parallel, there's more of like a force production benefit. But
Exactly.
when they're added in series, is there a force production benefit or not really, just the lengthening of the force?
Yeah, I guess there would be an indirect one. Like I said, you would be improving force production throughout the range of motion. But yeah, when you're talking about like the maximal force production that your muscle would generate at its optimal length, adding sarcomeres in parallel will be what benefits that more. I think about it as like if you have like one person pulling on a rope, like to move something, if you have like three people or like two people on either side of you that are also pulling it at the same time, that's going to obviously be able to produce more force and be stronger.
Yeah.
If you have three people in parallel pulling on something, that's going to increase the force. So yeah, that would be the parallel sarcomere analogy.
Okay.
Yeah.
Yeah, that makes sense. And for the stimulus that causes this, there's a lot of immobilization, or the
Right.
word immobilization. What exactly does that mean? Because I feel like people read that and they don't really understand what they mean by
Yeah.
that. And is it like static stretching or just holding a passive stretch?
Right. Yeah, so that's a good point. So there are immobilization studies that have also looked at sarcomerogenesis. So because sarcomerogenesis is regulated by muscle length in a way, there are studies that only in animals too, not in humans. When you immobilize the muscle in a stretched position, because again, you're placing those sarcomeres under that stretch stimulus, the muscle will add sarcomeres in series to sort of counteract for that. Even though like your muscles are mobilized, like that means it's basically your muscles in a cast. So like if you've ever broken an arm or anything like that, it would be like that. Except you would never probably cast a human muscle in a stretched position for any reason. It's usually just like neutral. But yeah, animal studies, they've immobilized the muscle in a stretched position. And because serial sarcomere number is regulated by the muscle's length, that results in addition of sarcomeres in series, even though like overall, the muscle's probably losing mass in that situation because you're not using it. So yeah, usually in those studies, you would see an overall decrease in muscle mass, but along the length of the muscle, an increase in overall serial sarcomere number. So yeah, if we look at passive stretching, that sometimes there's a few studies that would suggest that could increase sarcomere number, like sarcomeres in series, but never to the same extent or consistency as something like eccentric training, just because it's a way more intense stretch stimulus.
Interesting.
Yeah. There's one really cool study out there in mice. It was in like the late 90s, I think. But they use passive stretching to prevent the loss of sarcomeres during immobilization in a shortened position. So if we look at the opposite, yeah, if you immobilize a muscle in a shortened position, it's going to lose sarcomeres in series. Same idea. It's trying to keep that optimal sarcomere length constant to what it was before. But that's what that study did was for 1 to 2 hours a day, they removed the cast and stretched the muscle like fully. And that prevented the loss of sarcomeres in series that they usually saw with the mobilization in a shortened position. So passive stretching, I think, still has application in terms of like maintaining sarcomere number during a situation of muscle disuse. So
Yeah.
yeah.
All right. Well, then, would you say, or I'd say, like, can the same type of task that causes, sarcomeres to be added in series or sarcomerogenesis, can that task also cause sarcomeres to be added in parallel, per se? Like, can passive stretching cause sarcomeres to be added in
Right.
parallel, or can mobilization, eccentric contractions, etc.?
Yeah. Eccentric contractions, 100% can. So Martino Franchi, who I've worked with on a few papers, he's done that quite a bit. So I think that would have been from his lab. Yeah. So I believe they did this in humans too. So it was an eccentric training program, and they took biopsies of probably the vastus lateralis. And yeah, they saw that the cross-sectional area of the fibers for the biopsy was greater after eccentric training. So that would imply that sarcomeres were also added in parallel. Because if the cross-sectional area of your fiber gets wider, then that would mean that you've added sarcomeres sort of widthwise, not just lengthwise along the muscle. So eccentric training definitely can. Immobilization in a stretched position wouldn't, again, because like I talked about, you're still not using your muscle in that situation. So because you're not using it, it would undergo atrophy, and that would mean losing sarcomeres in parallel. Even though because sarcomeres in series is length dependent, you're adding sarcomeres in series, but you're losing sarcomeres in parallel. So yeah, those immobilization studies are an interesting case for like muscle contractile tissue. It's kind of interesting.
Yeah, and I mean, I brought it up because I spoke a little bit with. You familiar with Timothy Butterfield?
Yep.
He's done some work
Yep.
on this. I've asked him some questions, and he replied with immobilization, or like if we look at the old immobilization studies
Mhm.
done in animals on like guinea pigs from the past, like the
Yep.
1960s, something like that.
Yep.
There was an increase in force production, and he said that it could, like the increase in force could have been too big uh, for it to just to be caused by sarcomeres to be added in series. So it's possible that immobilization add
Yes. Okay.
sarcomeres
I
in
know what you're
series.
talking about. Yeah. Yes. So yeah, I guess some of the original ones, yeah, from Goldspink. That's right.
Yeah.
He did see an increase in force, or at least no difference in force. Yeah, with immobilization in a lengthened position. So yeah, I guess it is possible. It probably depends on the muscle and your model that you use for the immobilization. Because I did a study where I mobilized the muscle in a lengthened position. And yeah, I saw that it added sarcomeres in series, but like the muscle as a whole lost lost mass again, probably because it lost sarcomeres in parallel. But yeah, I guess maybe under a perfect scenario where you are able to like fully stretch the muscle and isolate it to that joint without causing any kind of shortening in any other way, then yeah, maybe it could still maintain its length.
Yeah, I mean, I
Or its force, is what I mean. Yeah.
he cited one study, but I know he said that there was a couple, and he just said like there's large mass and increases in muscle mass that
Mhm.
sarcomeres in series, which I do want to get into later.
Yeah.
Um, just wouldn't be able to cause. So I mean, maybe there are other factors. I don't know. Like you said, it could have been just the model or something they used that
Yeah,
caused
yeah.
that.
I think yeah, part of it is yeah, muscle dependent, or like there's more than one way that you can cast a muscle too. Like those old gold spink ones, I think they had a really good setup for their casting, that it was like fully isolated, like there wasn't anything else going on. That I couldn't quite figure out for mine. So yeah, it could have been some confounding factors that made it lose overall mass as well, that that wouldn't have been present necessarily for the older studies. So
Yeah, I mean, it's interesting to me because I know. I mean, I wouldn't be surprised if immobilization only caused increase in sarcomeres in series, but I mean, there is like some data out there that shows that maybe it doesn't. I don't
mhm,
know. I
yeah.
don't know if we have enough evidence to conclude, firmly.
Mhm.
But I guess now we can go into a little more nerdy topics
Sure.
about what are the mechanisms that could be responsible for
Mhm.
causing sarcomerogenesis. And is it just like things like passive tension, or if you want to go deeper about titin and the
Yeah.
extracellular matrix, or
Yep. So yeah, I think passive tension is a pretty big part of it. Uh, and yeah, I think Titan is probably one of the big like stretch sensors that would contribute to it. There's a lot of there's a lot of uh, protein kinases that are attached to Titan, and when you stretch it, those could unbind, because when Titan is stretched, it sort of changes its configuration, and that could set into motion any sort of pattern of molecular mechanisms that would regulate muscle growth. But yeah, as far as I know, because I did look into it quite a bit in one of the reviews that I wrote, it's a lot of the same, like when you get down to it, the same mechanisms that regulate just muscle growth in general. So like mTOR activation, things like down regulating mechanisms involved in protein degradation. So like there's a study out there that looked at the ubiquitin ligase pathway, and saw that down regulation of that might have contributed to the process of adding the sarcomeres in series. And yeah, I can't remember all of the ones that I found in that review off the top of my head, but yeah, a lot of them were
it's probably a
things that
lot.
regulated like protein synthesis for sarcomeric proteins like actin, myosin. But yeah, in terms of the actual stretch sensors, that's the really interesting question. I think Titan is probably heavily involved in that, but nobody's looked at it super directly. So
Yeah, I know there's Titan. I know some are saying that Titan is like for sure the mechanism, but I think there was like a lack of linking it to, was it mTOR1 pathway or whatever it was?
probably, yeah.
Yeah,
And
there's like
there's a
some
few other
missing
ones.
link.
So the integrin focal adhesion kinase complex is another one. Again, Martino has looked into that. And integrin is another structural protein that sort of attaches the sarcomere to the sarcolemma of the muscle fiber. So of course, yeah, when you stretch a muscle fiber, integrin's going to deform in some way that could put into motion a pathway that would regulate muscle growth. So yeah, there's all kinds of structural proteins in the muscle fiber that could be involved, and it's probably more than one too. I think it's because serial sarcomere number is regulated so tightly. There would for sure be more than one stretch sensor involved for it to be that precise. So yeah.
Yeah, and off the top of my head, I know there's some theories about ion channels. Is that
Yes. Yeah.
ion channels?
Yeah, so there's stretch activated calcium channels. I think I looked into that a little bit as well in that review. But I think there was one mechanism that some people have looked into where satellite cell activation might come about from that, and that could contribute to adding sarcomeres in series. Because satellite cells are like the resident stem cell in muscle. So in a situation where the muscle gets damaged, or maybe sarcomeres get overstretched and become damaged, satellite cells could be part of that repair process where it's making new contractile tissue, new sarcomeres to add along the length of the muscle. So yeah, activation of those potentially through a mechanism involving stretch activated calcium channels. I believe someone looked into that. Yeah, that could contribute. And there's
Yeah,
probably that they would too, because they're activated by
yeah.
stretch.
The other one on top of my head. Well, the ion channels one. I read something about like the whatever the ion channels release. Apparently, like I don't even know the word. It doesn't last long enough to cause any stimulus. It kind of just like
Fizzles out.
I want
Yeah.
to disappear, but yeah, dissociate quickly
Gotcha.
the molecules or the ions that are going through the ion channels.
Mhm.
I thought that was interesting. And then the other one off the top of my head was nuclear flattening. Are you familiar with that one?
Yes. Yeah. Yeah. I know what you're talking about. Yeah. So actually, I wanted to talk about that in my review, but not enough people had looked into it in the context of sarcomerogenesis. Yeah, when you stretch a muscle, the nucleus of the fiber also deforms and yeah, like flattens, gets stretched out. Because like so in the nucleus, that's where you would be transcribing the genes to create the like the contractile proteins. So yeah, of course, if you deform the nucleus, yeah, there could for sure be some way that it regulates addition of sarcomeres in series, definitely. But yeah,
Yeah,
not many people have looked into its involvement specifically in that process. There is a lot out there in terms of like nuclear deformation in regulating muscle mass in general or muscle gene transcription. But yeah, not in the tough context of sarcomerogenesis, unfortunately.
yeah, I've seen some of those. And I mean, going back a little bit with satellite cells, I've read and seen some papers that can kind of like reasonably indicate that satellite cells may be more, I don't know if active is the right word, but like more involved when it comes to like building sarcomeres in series than adding muscle mass or sarcomeres in parallel.
Yeah, possibly. I think I might have mentioned that in my review too. Now that I think about it, I should have read my review again before coming to the problem.
I try to read as many times as possible before
Yeah.
coming on here just to make sure I have everything.
But yeah, I think there was. I think you're right. There was something out there about that. There is a lot of controversy out there about how necessary satellite cells actually are for muscle growth in general. So there needs to be more research into that in general. But yeah, I think yeah, the idea that they could like how much they contribute to muscle growth in series versus in parallel is a cool question that could be looked into for sure.
Yeah, I mean, I know it's one theory, but I don't think it's like that's for sure what's happening.
Yeah.
Well, then, after talking about the mechanisms, uh, we talked about it occurring in animals. Is there a difference between sarcomerogenesis in humans versus animals? Like, are what causes it similar in both of them? And do we have any evidence for humans, or at least any direct evidence? I know I made like a tweet about this or an X about this.
Yeah. Yeah. So I would think that the mechanisms and like the stimulus behind it would be the same in animals and humans. Because yeah, like the building blocks of their muscles are exactly the same as ours. Like they have sarcomeres, all the same molecular pathways that are contributing to muscle growth in an animal are the same in humans. There's probably some minor differences that I'm not aware of because I'm not involved enough in like the molecular world, but yeah, in general, it's the same. So yeah, I would guess that sarcomerogenesis does happen in humans when we do something like eccentric resistance training. It's just unfortunately because measuring serial sarcomere number usually requires to like dissect a muscle out for like invasive measurements. That's why we've only really looked at it directly in animals. There is there is one way to measure, like a good in vivo way to measure sarcomere length, because in order to calculate serial sarcomere number, you need two things. You need fascicle length, which is like the length of a whole muscle fascicle, usually in humans measured by ultrasound, because then you can visualize it from end to end if you have a long ultrasound probe, and also sarcomere length, so that you can do fascicle length divided by sarcomere length to mathematically determine like how many times that sarcomere length would fit along the whole length of that fascicle. So in animals, we get sarcomere length by dissecting out a fascicle, and then usually we determine it by a method called laser diffraction. So that's where we shine a laser through the fascicle, and it produces like a diffraction grating as it passes through the A band, and the I don't know enough about the physics of it, but yeah. So basically, there's like a light region and a dark region in a sarcomere, and one of those it can't pass through, and the other part the laser can pass through. So it creates a diffraction grating as it passes through, where the light like diffracts in different angles, and depending on the length of the sarcomere, it'll diffract a certain angle, and then we can determine the length of the sarcomere when it gets picked up on our detector. I think I might have some pictures of it in one of my papers, but yeah. So obviously we can't do that in humans. We can't take their fascicle out and shine a laser through it. But there's a method that not many people have access to called microendoscopy, or it's often called the zebroscope, and it involves sort of like, I think I haven't used it myself, so I don't fully know how it works, but I believe it involves sort of inserting a needle that again it'll pick up basically the length of a sarcomere in the muscle that you're putting the probe on, and yeah, so you can use that again to get sarcomere length alongside you getting fascicle length with ultrasound, and then you could calculate serial sarcomere number. So there's a lab at the University of Queensland in Australia where they use that, and they did a study with 3 weeks of eccentric training, and they didn't see that serial sarcomere number increased using that technique, but they determined that it was probably because the training period wasn't long enough. Like 3 weeks, realistically, you don't usually see many like muscle morphological adaptations in that time for a resistance training program. And I actually saw this on Twitter the other day, but apparently they did the study again, but did 9 weeks instead. And apparently they did see an increase in serial sarcomere number. So this is like super recent data. That's kind of good timing for this podcast. So yeah, if that, I don't think they published it yet, but yeah, if it turns out exactly that way when they publish it, that would be like the first direct evidence of training-induced sarcomerogenesis in humans, which is kind of cool. But
Yeah, that was the Pinchera study, right? I think.
yes, exactly. Yeah.
I don't know if I
And
butchered
then I
that.
think
Yeah.
it was a different author on the newer one, but I think Pinchera is also on that one. Yeah, it's from Glenn Litchfark's lab in Australia.
Yeah. Dr. Jeff replied to my
Yes.
tweet about that, saying that like the evidence is indirect, but the best thing we have was the Pinchera study.
What's that one?
At 3 weeks.
Yeah. So
They
now
found
they've apparently followed it up with the 9-week study instead, which I would think that would be long enough to see those morphological adaptations, which apparently they did. So that's pretty cool. So yeah, again, I think it would happen in humans the same way as in rodents. And yeah, if their data is true and gets published, then yeah, that would sort of confirm that.
Yeah, and they use some sort of like state-of-the-art ultrasound. That's what
Yes, they
Dr.
also
Jeff said.
did I think they did 3D ultrasound, which yeah, that would be like the gold standard for getting fascicle length. Because yeah, I think you talked about on Twitter how fascicle curvature could also obscure some stuff, which it definitely can for human measurements. And 3D ultrasound will get around that because you could use like a segmented tool to measure the like full length of the fascicle instead of just extrapolating it. So yeah, I think they use that too. So it would be like super solid evidence whenever they end up publishing that.
That's pretty cool. I
Yeah.
actually didn't know that.
Yeah.
But yeah, there was that one, and then the 9-week one was the IBS abstract. I actually I saw it again on Twitter. I was scrolling through the tweets, and I guess one scientist say like, "What about this one?" And it was like just a low quality PDF, and I'm like, "This is interesting. I haven't seen this."
Yeah.
But would you say like that is sufficient evidence, or does it also depend on like muscles, because muscles have different neuromechanical behavior? I know muscles adapt differently sometimes.
Yeah, it can definitely depend on the muscle. I would say that yeah, like the evidence from that abstract is pretty solid evidence. Like I think it was in the hamstrings that they use. So the hamstrings have like really long do they have long muscle fibers? They're unipennated, so that would mean that when the muscle stretches, that could, I guess, buffer the fascicle stretch a little bit. But what am I trying to say here? Yeah, so basically, when you stretch the hamstrings, there is quite a big change in fascicle length that happens. So yeah, it would make sense to me that if you do, they did like Nordic hamstring or eccentric hamstring curls. Yeah, it would make sense to me that if you did a super intense eccentric training program like that with that muscle, it would add sarcomeres in series. But so in rodents, we don't always see an increase in sarcomere number in every muscle. I think some people who have tried it in like the TA. No, because in rabbits they used the TA, and it was like the tibialis anterior, and I think they saw it in that too. But there's some muscles where we don't always see it as much. Maybe the EDL is what I'm thinking of, compared to like the soleus, the vastus intermedius, the tibialis anterior. Those are usually the ones that are used, and I guess the gastrocs as well in animal studies. But yeah, I don't think the EDL has added sarcomeres in series as much as those other muscles. Or I might be wrong about that off the top of my head, but yeah, it is muscle dependent for sure, and I don't know what exactly affects that. Some muscles just seem to be more attuned to it than others.
Yeah, I want to get into that later on, because there's one theory going around social media that's just being thrown around like crazy that I do want to get into. But finishing off with this question here, I know in the Pinchera study they didn't find an increase in sarcomere addition in series, but they did find the increase in like the sarcomere length of itself.
Yeah.
do you think there's like a time lag for that?
It's possible, yeah. So what they saw, because they measured fascicle length and sarcomere length at the same joint angle pre and post training. So yeah, basically what they saw was that fascicle length was longer at that joint angle post training compared to pre training, but it couldn't be attributed to more sarcomeres in series because sarcomere length was also longer at that joint angle post training compared to pre training. So like mathematically, serial sarcomere number was the same when they did the math. So yeah, there could be some kind of timeline thing where, and actually there's been studies that have looked at that using like transmission electron microscopy, where you can actually like visualize the sarcomeres, where yeah, you see that initially the sarcomeres, as they're getting overstretched from whatever stretch stimulus you're using, they probably stretch to a certain point and then maybe split, and then could become like two new sarcomeres from each of those halves. Again, there's a little bit of controversy out there in terms of how that happens and like where along the muscle that it happens, but I feel like I've seen evidence that's convincing enough that that could be how it works. Where yeah, it's a timeline thing where sarcomere initially gets longer, it splits, and then you grow it from there, basically.
Yeah, I mean, it's interesting why it would do that. I don't really know. I wish I had an answer to that. But another piece of evidence that I see people use is that, well, we know that we've seen changes in like peak torque angle, and so how can you explain that if sarcomeres weren't added in series?
Right.
I know I asked this to Dr. Jeff, and he replied with that there's other factors that can affect that, like stiffness of the muscle tendon.
Yeah, increased compliance. Yeah, so that could affect it, I guess. So basically, I think what he was getting at there is if so basically, because the muscle has like also the tendon and the connective tissue that sort of wraps around, well, the tendon doesn't, but the connective tissue wraps around all the muscle fibers and then converges onto the tendon before it attaches to bone. So there could be a case where if that connective tissue and the tendon become more compliant, that would mean that you can stretch it more without producing like a big increase in stiffness. Then yeah, there could be a case where you could stretch the tendon and the connective tissue more before the actual contractile tissue in the muscle gets stretched to the same extent. So in that case, you could stretch the whole like muscle tendon unit or the joint, and the joint angle would be technically longer, but the muscle with the same number of sarcomeres in series wouldn't reach optimal overlap until it's at that more stretched joint angle, because the tendon and the connective tissue are able to stretch more because they're more compliant. So yeah, I don't know. I have no idea how you would like tease apart those two different influences. I guess yeah, like measuring actual sarcomere number would confirm that's the case. But yeah, I think in general, when you see an increase in the optimal muscle length, it's probably because you've added sarcomeres in series. But yeah, there's some confounding factors that can affect it for sure. Like
yeah, it's still indirect evidence just because of
yeah,
pre and post stiffness from
exactly.
The intervention or whatever.
Mhm.
So yeah, I mean, I've been told it's like, well, can you explain this? And I'm like, I mean, I don't know if it's pure from sarcomeres in series. I would assume so, but I feel like there's
Yeah.
other factors. And, well, I asked on Twitter and I got a pretty good response, which was interesting. So I know the next question was actually going to be about the difficulty of measuring sarcomere length, which I think we talked about a little bit, where it's like it's pretty invasive procedure and not a lot of labs in the world have it.
Yeah, so that technology I talked about, the Zebra scope, is, I forget how much it costs, but yeah, it's super expensive, and I don't think they even make them anymore. So yeah, like the labs that have them right now, those are the only labs that have them and will have them for the foreseeable future. Yeah.
Do you know which labs have them or anything? Because I know
So,
people would like
Glenn
papers.
Lindmark, yeah. I think Scott Delp, is that who it was? And What was the other one? Okay, one sec. I'm just gonna look something up on my.
I don't know if you heard about Andrew Vygotsky. He told me that Northwestern, I think,
Yes, I
has.
think that's what I'm thinking of. I just have to double check where this paper is from. Uh, Wendy Murray. Yes, that's what I'm thinking of. So yes, she might be at. If that's if that's where I'm thinking of, but yeah, Wendy Murray's lab. They did a study in stroke patients where they looked at serial sarcomere number in like the affected, like the limb that was affected by the stroke compared to the limb that wasn't, and they saw loss of sarcomeres and series in the limb that was affected by stroke, because it was like sort of stuck in a shortened position, basically.
Yeah,
So
and
yeah,
well, there's also I replied to you about this about trigonomic equations, how there's some of them, but and this was actually from an article by I don't know if you heard of Stronger by Science. They talked about some things about like measuring sarcomere length is pretty difficult, and there's trigonomic equations, but they kind of assume that the fascicle is like linear.
exactly. Yeah.
And hypertrophied muscle is curved and all that, but you said that's not necessarily a problem. There's a bigger problem than
Yeah,
that.
yeah. So definitely using the trigonometry is not like the most ideal way to measure fascicle length. It's because most ultrasound probes are um, like 10, 20 centimeters. I guess, yeah, probably not 20, like 10 centimeters long, which is obviously not as long as a fascicle would be, right? So you usually can only visualize like where the fascicle inserts onto the aponeurosis and then not all the way to where it inserts on another aponeurosis. And then, yeah, you have to use trigonometry with like the pennation angle and the thickness of the muscle to sort of extrapolate like where it'll end up. But yeah, there's other techniques. There's extended field of view ultrasound, which basically what that does is it puts two shorter ultrasound probes side by side and then sort of like combines the image together overall, and then you can visualize the whole fascicle from end to end, or 3D ultrasound, like I talked about earlier, where you can really track like its entire length. So yeah, trigonometry, the trigonometric way is definitely not 100% ideal. There's studies that have like looked at its reliability and validity, like compared to those better methods where you can actually measure it from end to end, and apparently it's accurate. But yeah, it's a good way to do it if you have no other options, basically. But yeah, the bigger issue, like you were getting at, is that we can't really measure sarcomere length in humans in vivo unless we have that microendoscopy equipment.
Yeah, and I had to write this down. You replied to me also saying that like we have a bigger issue other than the trigonomic equations because um, we have no measurement of what sarcomere length is in general, so we can't
Yeah.
necessarily like do sarcomere.
Yeah.
Was it sarcomere?
So basically, this is sort of like what the Pinchera study demonstrated. But yeah, because fascicle length can change depending on the joint angle that you're measuring it at. But serial sarcomere number would be the same no matter what joint angle you're measuring it at. You can't fully rely on just fascicle length as a measure of serial sarcomere number. The one way that some studies have gotten around it is measuring fascicle length at the optimal joint angle, because that could assume that like that's the optimal fascicle length, and then there's like a predetermined optimal sarcomere length in humans that then you could use to estimate serial sarcomere number using that equation where you do like optimal fascicle length divided by this predetermined optimal sarcomere length. So yeah, that would be like the next best way to do it if you don't have microendoscopy. That's like your best guess at the serial sarcomere number in humans.
Yeah, and I sent you a paper that I'm gonna be honest, I didn't really fully read it. I kind of skimmed through it.
I skimmed over a lot of the mathematical modeling stuff that I don't know enough about.
Right. I mean, I'd like to know. I'd like to understand that math. Hopefully. But yeah, you told me it was mostly about when using fascicle length to assume serial sarcomere number. what was it? What did they do again? It was in rabbits. They did models.
Yeah, so they use mathematical modeling to sort of guess at the time course of like the sarcomere length um, adaptation and like the sarcomere addition, sort of like what we were talking about earlier, where like the sarcomere might stretch first and then sarcomeres could be added after. Um, yeah, they sort of use mathematical modeling to like predict the time course of that. If we could somehow apply that same mathematical model to humans, yeah, we could guess at like what sarcomere length would be in a human scenario to guess what serial sarcomere number would be from fascicle length. So yeah, it's kind of cool. I'm surprised I hadn't found that paper before you sent it to me. Seems
I
really
don't know how I found it. I sometimes just stay up late at night and just
yeah.
randomly put words together, Sarcomere number, measurements, or whatever, and then
Yeah.
every time on Google, like a different paper shows up, and I saw that one. I'm like, I haven't seen this one before.
Yeah.
And I mean, it's still like early development of it. The equation has been like a big discovery or something.
Mhm. But
was it? Yeah, it wasn't like an equation where it's like, okay, like this is the new thing, or this is like a gold
yeah, it
standard
was
thing.
like a step in the right direction in terms of trying to develop that in humans. I think that was like the main like clinical application that they talked about in their paper.
Yeah.
But yeah, I think again, I don't know enough about mathematical modeling, but I feel like there's probably a way to try to extrapolate that to humans in some way. I would think, or at least like make the best guess about it, I guess.
It'd be nice. It'd be
Yeah.
nice. So I guess moving on from the mathematics and trigonometry, is there a limit to sarcomerogenesis? Because
Hmm.
I've seen some data that it occurs rather quickly, at least
Yeah.
more so than sarcomere being added in parallel.
Hmm.
However, the data does seem inconsistent, and I feel like there's a lot of other factors. So I don't know if like there's a limit to that. And I
Yeah.
mean, obviously, muscle can get longer forever.
Yeah. So that's pretty much, yeah, that would be my answer. There's definitely a limit. Because yeah, like eventually you'll reach a certain point where like you can't apply any further stretch stimulus on the muscle to make those sarcomeres get overstretched. So yeah, once you've reached that limit of like um, the full, like you've stretched the muscle so much, you've added the sarcomeres to reduce that stretch, like the tension stimulus. And yeah, then you won't be able to stretch the muscle any further because yeah, there's a limit on our joint range of motion that would prevent us from further stretching it to provide a further stretch stimulus to add more sarcomeres in series.
Yeah, and I guess the more difficult thing comes with like the specific timeline that it plateaus because was it Blavovic's
Yeah.
lab something showed one about like six weeks or something, but again, it seemed like assumptions because they don't have those invasive procedures. So there
Yeah,
was like a lot
exactly.
of
Yeah, it
speculation.
would have been just using fascicle length for like assumed sarcomere number. Yeah, it does seem like it happens quicker, like you said, than addition of sarcomeres in parallel, possibly. I don't know exactly why. I think part of it is because, um, again, I think just the like muscle length stimuli are just more sensitive than the stimuli that would control sarcomeres in parallel. Like obviously we can like our muscles range of motion is pretty broad. So yeah, it would make sense that it could be sensitive to even like small changes in muscle length. So yeah, it seems to be like that could be why. And maybe that's why it can adapt so quickly as well. Just because, like I talked about, there's so much that could be regulated or that could be sensing that stretch in a muscle fiber, all the different cytoskeletal proteins. So yeah, I have no like Jeff and I talk about this all the time, like why it adapts so quickly and why it's seems to be quicker than sarcomeres in parallel. But yeah, that would be my guess. It's just maybe the muscle is more sensitive to stimuli that are associated with a change in muscle length compared to what would be driving addition of sarcomeres in parallel.
Yeah, and I mean, it's hard because I feel like there you start kind of stepping into a little bit of like anthropology and like why they were designed or
Yeah,
like why
exactly.
they evolved that way.
Yeah.
And that's one of the reasons why I love and hate like the field of exercise science, because it's
Mhm.
like it's hard to become like an expert in everything. It's either you spend all your life focusing on one thing, but then the next thing you know, that thing can like spread into so many different
True.
topics and subjects. It's like if we
Yeah.
lived 300 years, probably. But
Mhm.
what was I gonna say? So on top of that, and why they occur quickly, do you know like a specific like time date? Is there like a firm time date? Like, oh, sarcomerogenesis usually plateaus between 6 to 9 weeks.
Yeah, yeah.
Is
So
there
again,
really?
in animals, we know it really well. So in animals, I think some people showed it happened in like 2 days. In rats, like 2 or 3 days. That was with the like immobilization in a stretch position model. So yeah, like in rodents, it seems to happen super quick. In humans, I would guess it's a little slower than that because like we have a slower metabolism than rodents, so things just get turned over a little more slowly. I guess if we go off of that, those two Pinchera studies, the 3 week one and then this 9 week one that was that conference abstract, I guess it must take longer than 3 weeks, clearly, but less time than 9 weeks. Because I think the 9 week one again, if that abstract gets published, which I hope it does, because it seemed pretty cool. I think they saw like a 50% increase in serial sarcomere number, which is like that's a huge amount. So if the increase was that large in 9 weeks, I would imagine it would have started to happen much earlier than the 9 week time point. So yeah, it could have even started like if the Pinchera study had done 4 weeks even instead of 3 weeks, maybe they would have seen it. So yeah, somewhere beyond 3 weeks, but also much earlier than 9 weeks. If I'm going off of those two studies in humans, that would
Yeah, I mean, the data at least looked a little bit inconsistent sometimes. I don't know if there's one that lasts like 32 weeks, but I don't know if it was specifically sarcomerogenesis. I don't know if it was just like how
yeah
their methods or anything. Um, but I assume part of it could be like to decrease in passive tension, maybe,
yeah
because obviously when you stretch the muscle, assuming all things remain the same, and I think
mhm,
that's where it get complicated.
mhm,
Uh, the muscle would decrease in passive tension, a little bit
yep
of passive tension, um,
yep
and then therefore it could reduce a stimulus maybe.
exactly yep. Um,
Um, However, would you say like there are factors? Because like I said, assuming all things remain the same, like when
mhm,
we grow, there's changes in pennation angle, and
yes yep
there's things that can increase the packing of fibers, and
yep
so it allows more. And I think muscles love a bit of passive tension, so I don't think it ever gets to zero. I think there's like a sweet spot.
yeah
But I don't know what you think of that.
yeah. So yeah, you mentioned pennation angle. That's like one of the biggest confounding factors that could interfere with it for sure. Because yeah, like your muscle has a mechanism called muscle gearing, which pennation angle contributes to, where during stretch or shortening, the pennation angle allows the fibers to potentially rotate such that like the whole muscle might be shortening or lengthening, but like those fascicles could just be rotating like that and not actually stretching or shortening along their length, because that's what pennation angle exists like that. Again, it's an evolutionary sort of thing, to potentially protect the muscle from those drastic, or protect the individual sarcomeres from those drastic length changes. Um, so yeah, so pennation angle can interfere in some way with the addition of with like how much we might see the addition of sarcomeres in series. And the other one I talked about earlier is the connective tissue, because when we expose our muscles to exercise or mobilization or any of these things we're talking about, the connective tissue also adapts alongside the actual contractile tissue. So depending on what happens with that, that could also interfere with what we see in terms of structural adaptations and also the
Yeah,
function impact,
and the what? Sorry.
and like the impact on function, like muscle function.
Oh,
Yeah.
uh, yeah, and there was a good paper. I think it was from 2015. I don't remember who wrote it, but it showed like increases in pennation angle I don't know if it happens through immobilization or like through passive stretching. Can that occur?
Yes. So usually with immobilization, you see a decrease in pennation angle again, because so the other thing that pennation angle could be associated, that you would see it associated with, is sarcomeres in parallel, because when you pack more sarcomeres in parallel, your pennation angle basically allows you to do that. So as you increase the pennation angle, Jeff sort of describes it as like if you're packing cards under your hand. So like picture me packing like more and more cards under my hand as I lift it up here. Like as I increase the pennation angle, I'm packing more and more cards in parallel on top of each other. So if you can picture that as like pennation angle increasing is a way to basically fit more contractile tissue in parallel, which is going to allow you to generate more force in like a given size of a muscle. So yeah, the way that it changes can interfere in some ways. And yeah, so yeah, we were talking about immobilization. So yeah, basically immobilization, you're not using your muscle. Because you're not using it, you're going to be not generating force. And because parallel sarcomere number is what's associated with like your muscle's maximum force production, you're going to be losing sarcomeres in series, or sorry, not series, sarcomeres in parallel with any or most immobilization, I guess, maybe not stretch like we talked about earlier. But yeah, with immobilization in general, you're going to lose sarcomeres in parallel, which would probably show up as a decreased pennation angle in an ultrasound image. So, and yeah, we've seen that in our lab too, with the rat studies that we've done.
Yeah, cool. Yeah, there's like a lot of I don't know if I guess inconsistency is the right word, but at the same time, it's like, you know, sometimes one study would show this, and then another study would show increases or decreases.
Yeah.
It's like
that's one of those things that I think is muscle dependent too. Yeah, I think it all depends on like what the muscle. Probably what it comes down to is like what was the muscle designed for, again, evolutionarily, that like what are you then changing about what that muscle is doing for a given, whether it's immobilization or training intervention, that will contribute to how that stimulus impacts the way the muscle will adapt. So yeah, I think there's probably some muscle dependent effects contributing to that, to those inconsistencies depending on the study.
Yeah, I mean that is a good reasonable explanation. I think what could be happening there. So now, I mean, now people have been applying this sort of not reasoning, I guess. It's like, okay, well, sarcomeres have a certain, or muscles have a certain adaptation of adding sarcomeres in series. So can resistance training cause sarcomerogenesis? I know you talked a little bit about eccentric versus concentric adaptations. If you can go a little bit deeper into that.
Yeah.
And is the stimulus, actually, I'll ask that later. But yeah, does resistance training occur in they're called sarcomerogenesis? I know you sent me a paper on that.
Really yeah, so I think resistance training probably results in adding sarcomeres both in series and in parallel. Because yeah, you have both like conventional resistance training, you have both that concentric phase of the lift, but then also if you're resistance training properly, I guess, you're also focusing on that eccentric lowering phase where you're controlling the descent of the lift as well. So yeah, I think conventional resistance training probably results in addition of sarcomeres both in parallel and in series. And actually, so the indirect way of measuring sarcomeres in series that I talked about earlier, where you measure fascicle length with ultrasound at the optimal joint angle. There was a study by Neil Reeves in 2004, I think, from Marco Nerici's lab. I think that's where it was. Yeah. And they did that with the resistance training program, actually in older adults. And if we go off of that, they saw what could be assumed as an increase in sarcomeres in series with just conventional resistance training, where you do both a concentric and an eccentric phase of the lift. So, and I think that was
I mean.
I
mean, when you're doing a bicep curl, you're doing concentric, eccentric. So yeah, exactly. I mean, you could say that like the concentric also can cause the addition, or is the reason why you see addition of sarcomeres in parallel, because concentric
Exactly. Yeah.
contractions, and eccentric, you could say sarcomeres added in series.
Exactly. Yeah.
Or at least eccentrics have like more of a eccentrics have like a preference of adding sarcomeres in series than adding them in parallel. But would you say it's like if we were to just do like forgot the word for it, just a pure eccentric contraction with no concentric, would you still think that that results in sarcomeres in parallel because there's a force generating component?
Yeah, I think it can. Again, it probably depends on the muscle because, like I said, I think Martino did see like an increase in fiber cross sectional area with eccentric training, so that would suggest that yeah, it could result in sarcomeres in parallel. I think what it probably comes down to is concentric alone would cause a bigger increase in sarcomeres in parallel than eccentric training would, but I think eccentric training still could cause an increase in sarcomeres in parallel because yeah, like you said, there is still like you're producing a strong force aspect of it, which could contribute to some kind of stimulus for sarcomeres in parallel. So yeah, I think
Yeah,
maybe not quite as much as the concentric aspect.
yeah, and so I guess would the stimulus be different from static stretching?
Mhm.
Because I emailed you about this too. It's like, why could it be that like muscles grow more at longer length? Because is it
Mhm.
just because it's just stretching component, like a force plus stretch that causes this
I
kind of
think
thing?
that's what it is. Yeah. I think there's probably both an active and a passive component to it. Where yeah, like you're putting tension in the muscle just by generating active force. But then when you add on top of that the passive force that comes with stretching your muscle, um, there's probably an aspect of both of those, I think, that would go into the like the molecular response that drives the muscle growth, specifically adding sarcomeres in series. Yeah, and again, we don't
Yeah,
know many of the like very specific mechanisms of how that works, but yeah, in general, it's driving muscle growth in some way, making it specifically add the sarcomeres in series.
yeah, and I mean muscles. I mean there's a good paper by it was like mechanotransduction and skeletal muscle or something that essentially like
Yeah.
they grow from like deformation,
Mhm.
like that causes this stimulus of mechanotransduction.
Exactly.
I guess one theory that I've seen a little bit or heard a little bit around is that like muscles are more vulnerable to that deformation in a more lengthened position.
Mhm.
Maybe that could be a reason why, or they're more the mechanical transduction is more perceived and like that force induced strain in the lengthened position.
Yeah,
We'll
I think
say.
that makes sense. Yeah, I guess yeah, you could look at it as like you get more um, like a maximal activation of all these different mechanotransduction pathways by yeah, generating force in the lengthened position compared to if it wasn't in a lengthened position, or if it was in the lengthened position without generating active force as well. Because yeah, I think so.
Yeah, and
That's a good point too. So some of those older studies that were in like rats and mice and rabbits, they immobilized the muscle in a lengthened position, which we know leads to sarcomeres being added in series. But they also they had another group where they applied electrical stimulation to the muscle on top of that, and they saw that that caused it to add even more sarcomeres in series. So that would be like the passive component and the active component combined together. So yeah, that would kind of go along with exactly what we were just talking about.
that's pretty cool because I've only seen that I know I've seen where like they cut the nerve and it's like the muscle
That one too,
doesn't
yeah.
they don't really need to be active to sarcomeres in series or grow when they're passively
Yeah,
stretched.
yeah, so yeah, in that case, they've cut the nerve, so that would limit it to just the passive component. And yeah, they still saw sarcomeres added in series. So pretty cool ways that they manipulated all the different stimuli, I guess.
Yeah, and I wonder why. I know when we talked about mobilization, you said that you saw decreases in sarcomeres in parallel. But when we passively stretch a muscle, we've seen increases in muscle mass and force, possibly due to sarcomeres being added in parallel along with sarcomeres in series.
Right.
Why do you think that can be if we're both kind of like stretching the muscle? Or it's like, when people think of immobilization, like, oh, well, I'm stretching my muscle in this position for a while. Like, why is that not the same thing as passively stretching it?
Yeah, I don't know exactly. Passive stretching is interesting because uh, a lot of studies, like when you do like really intense passive stretching interventions, oftentimes you'll see an increase in joint range of motion. And some people have looked into whether that could be due to an increase in sarcomeres in series, which like that would make sense. Like adding sarcomeres in series, you should be able to stretch your muscle farther before you get that increase in passive tension that prevents it from stretching more. But a lot of them have seen that sarcomeres in series, or I guess fascicle length, and the ones that did it in humans didn't change. And it was more associated with like a reduction in or an increase in pain tolerance, or not necessarily increase in pain tolerance, but like decrease in pain associated with stretching your joint to a certain like extent of the range of motion. So yeah, we don't know too much about whether like static stretching. I think again, it probably depends on the intervention or the muscle. If static stretching will be increasing sarcomere number, or increasing joint range of motion from some other way, such as reducing the pain associated with stretching the joint.
Yeah, would you say there's like more tensile force when we passively stretch than
Yeah,
immobilizing or anything like
yeah.
that?
It seems like that's what it is. Yeah, versus like these immobilization studies in the rodents, where yeah, I think it just comes down to like this muscle is fixed in this lengthened position for like weeks, basically, and that just causes it to regulate the sarcomeres in series because of that. Because the muscles can't think, obviously, but if the muscle was thinking, it would be like, oh, I got to add some sarcomeres in series because these sarcomeres are all way overstretched and they're just stuck here. Um, so yeah, in that case, yeah, it seems to be different from static stretching. There is a way
Yeah,
that mobilization studies have been applied to humans in orthopedic settings. So there are cases where like people are born with one limb that's longer than the other, so they can cause the limb that's needs to be longer to get longer by sort of incrementally lengthening the bone, which obviously sounds super painful, but there's like surgical ways that they do it safely. And that causes, if you do it very slowly, incrementally, that causes the muscles that are attached to that bone to add sarcomeres in series so that the limbs then become equal, both in terms of their overall length and their muscle length. So that's the way that those immobilization studies, I think, are best applied to like human populations.
yeah. There's a lot of like physical therapy with this,
Yeah.
or like a lot of combination, or layover. I would say like with immobilization, maybe it could also be like they don't when they mobilize the muscle, it's not like not in such a position where there's like a lot of tensile force. Maybe because I think it was high intensity stretching does seem to cause this increase in sarcomere
It's
numbers because
very
it's not
intense.
that.
Yeah. Usually they do see that. Yeah.
Yeah, so if it's not that intense, I guess if it's mobilized in not such an intense way, then it could
Yeah.
possibly be leading to why there's not an increase in force.
Yeah.
Um,
Yeah. That's true.
but that does something I do wonder is if sarcomerogenesis can cause increase in muscle cross sectional area. I know
Mhm.
we talked about it, but I do want to come back to this. I've seen some models from Hornberger lab. I don't know if
Yes.
I'm
Okay.
pretty
Yeah.
familiar.
You're probably talking about like they have like drawings, right? So you're talking about?
Yeah, I think it was on a guinea pig or something.
Yeah. Yeah. Hornberger Lab. Yeah. I think we're thinking of the same one. Yeah. So yeah. So this would be in the case where again, like pennation angle makes stuff kind of um, weird, I guess. So yeah, like if you have a muscle and the fibers are so like this is think of this as like one side of the muscle, the other side. If the fibers are out on an angle, like going from one side to the other. Um, and you are increasing the length of that fascicle, so adding sarcomeres in series, then because it's on an angle, then if you make it longer, it will cause the cross-sectional area or the width of the muscle to get wider, just from adding sarcomeres in series. So yeah, in cases like that where the muscle is pinnated, which most muscles are in the human body, then yeah, you could increase the whole like cross-sectional area of the whole muscle, both by adding sarcomeres in parallel, but also by adding sarcomeres in series. So
Yeah, and I mean,
yeah,
it's
that's
also
one of those ways
possible.
that like pination angle makes things mathematically just hard to visualize, I guess.
Just
But yeah,
really dirty.
Hornberger has done a good job drawing that out in some of his reviews. It's kind of cool.
Well, that's still like I don't think that's still like a confer like a lot of affirmity there, right? Because it's also possible for a muscle that sarcomeres in series and not decreases in cross sectional area, I assume.
Yes, yeah. It would be purely from like a mathematical scenario. Yeah, in terms of like whether or not you actually see that in the real world following a resistance training program. Yeah, there's probably a lot of inconsistency out there. Again, depending on the muscle or the intervention, like what the stuff we talked about.
Yeah, and it was a model. I think it was sent like to like the editor or something. That wasn't necessarily like a paper.
Okay.
I don't know if that was. I've seen something about saying like it was a letter sent to the editor. It wasn't like a finalized model.
Gotcha.
The model worked in some papers, didn't work in some other papers.
Yeah.
So I was just curious about it. I tried asking him, I didn't get a response.
True.
But I mean, they're probably busy as well. So
yeah.
I guess that leads to the final question for a quick Q&A. If all muscles can experience sarcomerogenesis, and we kind of talked about this throughout the live, like you said, we say it depends on like again the type of muscle or anything like that, or.
Yeah. So I think every muscle, yeah, again, because like the same like fundamental structure and molecular pathways are going to be existing in any muscle. So every muscle, I guess when you think of it like that, should be capable of sarcomerogenesis. But yeah, because like there are different like origins and insertions in muscles that could change like between different muscles would change like the length stimulus or the strength of the length stimulus that could be applied to it. Yeah, so technically it should be possible, but just because of the way that different muscles are designed and what their function is in the human body, that would probably influence what extent you'd be able to induce sarcomerogenesis with like a training intervention in those muscles. So.
Yeah, so I bring this up because this is kind of like the final thing that everyone's kind of like confused about, where there's this theory talking about how a muscle can only experience sarcomerogenesis if its sarcomeres can reach into the descending limb of the length tension relationship. If it doesn't do that, then therefore it can't experience sufficient passive tension to experience sarcomerogenesis.
Yeah,
I have always been a little pushy about that theory. I don't know if.
I think it makes sense. Like if like what we talked about earlier, like the main stimulus for sarcomeres being added is those sarcomeres getting overstretched. So when they're getting overstretched, that's when they're going to be on the descending limb of their force length relationship, where those actinomyosin can't bind as much because they're just stretched further apart from each other. So yeah, to me it makes sense that that would be a pretty necessary step for it. But yeah, there could be other stimuli that could regulate it. Like there are like stretch of connective tissue. We've talked a little bit about connective tissue. There are some molecular like responses in muscle that are regulated by connective tissue stretch as well. So, and yeah, like we talked about even like stretching of the nucleus. If in some way that could happen without the sarcomere also being stretched, then yeah, maybe that could regulate it in some way. But yeah, I think in general, the sarcomere does have to get overstretched in order to stimulate sarcomerogenesis, because that's when you're going to be stretching tighten as well, when you stretch onto that descending limb.
Yeah, I know. I forgot the name of the researcher who sent this to me, but he's like, he replied again on one of my Twitter. That we have seen that, and I think this is talking more not necessarily about passive stretching, but about like resistance training at longer lengths.
Mm-hmm.
Like the sarcomeres don't necessarily have to be maximally stretched, just relatively lengthened enough to experience that sarcomerogenesis or that extra growth.
Yeah. So that's a good point. Yeah, they don't necessarily have to be maximally stretched, but yeah, maybe even just stretched more than what they normally are. So yeah, I guess there could be a scenario where like if the muscle is for whatever reason usually on its ascending limb, and that's like its normal, that's its normal like functional place that it's normally at, and then you were to stretch it onto like its plateau region of its force length relationship, maybe that could be enough of a stimulus to put it back to what it was originally at. So yeah, again, yeah, that's another. It could very well all be muscle dependent when it comes down to like the real world in the human body.
Yeah, I mean, I definitely think it's. I mean, it's logical. But again, like you said, there could be other factors because there are like weird muscles like the gastroc where I think it's on the ascending limb, but we still see it grow very well from like being passively stretched.
Yeah.
So I think there could be other things happening there. There's like variations of tighten also, and I think what was it? The elements that produce passive tension could be lengthened to different extents than the sarcomere. So like, let's say the sarcomere doesn't experience or doesn't reach deep into the descending limb. Maybe there's like passive elements that have
That's true. Yeah,
like,
yeah. There are other passive elements outside of the sarcomere could be changing length somewhat independently for sure. Yeah.
Yeah, like you have a lot of connective tissue, and I know there's like contention about tighten and the extracellular matrix on passive tension. Rick Lieber does a lot of good work on that.
Yes, he does. Yeah.
And
a lot of
Thomas Day. Yeah, and then I recently read this paper. It was pretty hard to read. I was looking at a lot of, but it was a good paper where I think his name was Thomas J. Roberts. He laid out like the evidence of tighten and the ECM objectively, just to see where the research is at. So I think that's pretty cool because if it's just tighten, I mean, maybe the
ECM, like the ECM is what contributes to most of the passive tension in our muscle at the whole muscle level. Like when you get to the level of the whole muscle, tighten does contribute just like a very small amount. So I think tighten is more involved in like the regulatory mechanisms. Whereas like the actual passive tension that we observe in a muscle is more going to be about like the ECM, which is connective tissue converging onto your tendon and all that. So.
Well, when you say like ECM is more involved with tight or like it bears more passive load than tighten, like we haven't really seen any loads from tighten that match loads on muscle. Would you say? Like if there are changes in tighten and helping passive tension, like it's probably going to be very minimal.
Yeah, pretty much. I think there's a review out there. I forget who the author is, but it basically compiled passive force data across a bunch of different species at like each different structural level. So like all the way from like single muscle fiber where there's no connective tissue. So the passive force there would mainly be due to tighten. And then they had like fiber bundles where there's connective tissue between the fibers, and then fascicle, whole muscle, and then joint level. You see like way steeper increases in passive force as soon as you start adding connective tissue into the mix compared to when it's just tighten. So yeah, it's definitely more the ECM when you look at passive whole muscle.
That's interesting. I'd have to look for that. I haven't seen that.
Yeah, I think the author might have been Ben Binder Markey. I could be wrong about that, but I think
Bender Markey, something like that. He worked with he had a paper with Richard Lieber again on measuring fascicle length. I think it was like on the gracilis or something. I don't remember.
Yep.
But essentially, like the data is confusing.
Yep.
On measuring fascicle length. But yeah, he does a lot of good work from what I've seen, at least.
Yes, definitely.
So yeah, I mean, those are pretty much all the questions, and I guess we could finish off with a quick Q&A on some questions that people want me to ask you.
Sure.
The first one being, if you know what type of fibers or muscle fibers are added through sarcomerogenesis, like which fiber type, if it's like type 1, type 2. I've seen some evidence of both.
I don't have like a concrete answer on it. I guess it might depend on like what the normal fiber type proportions are for like a given muscle, because muscles have different proportions of fiber types. Like the soleus or any muscle that's heavily involved in postural control is more slow type usually, because they need to be less fatigable. So yeah, in that case, I would expect that the sarcomeres that are getting added would be more like slow type, or sarcomeres for slow type fibers. So yeah, I guess it would depend on the fiber type proportions for a given muscle, I would think.
Yeah, I've seen some evidence. I've mostly, again, I've seen evidence that it's usually like type 1 fibers being added. But then there's also some evidence, or I wouldn't say some, maybe there's other evidence, or just as much showing like type 2 fibers being added. So it's
because I think type 2 fibers are more susceptible to muscle damage during eccentric contractions compared to slow type fibers. So so if those ones are getting more damaged and getting more of that stretch stimulus, then those might be the ones that are getting prioritized in terms of adding sarcomeres in series. So there's probably a few different ways to look at it, I think.
That possibly could be one mechanism. Now, another question was, is sarcomerogenesis the same as stretch-mediated hypertrophy? I don't know if you heard that term.
Yes, I have. I guess so. Hypertrophy, I guess, refers to like muscle growth as a whole. I think when we think about hypertrophy, it's usually thought of in the context of adding sarcomeres in parallel, like increasing fiber cross-sectional area. But I think in that context, they could also be talking about it in terms of adding sarcomeres along the length of the muscle as well. So I guess stretch-mediated hypertrophy could encompass both adding sarcomeres in series and in parallel in response to a stretch stimulus.
Yeah, because again, Stronger by Science recently came out with an article talking about length and partials, like just training muscles at a longer length in the partial position. Don't cause stretch-mediated hypertrophy, because everyone's just throwing the word stretch-mediated hypertrophy, saying like, if your muscle grows more at longer length, that's stretch-mediated hypertrophy. But then they make the argument saying, well, not really, because stretch-mediated hypertrophy is specifically talking about immobilizing or passively stretching the muscle for long periods of time. Or in resistance training, maybe it's not the stretch that's augmenting the growth. Maybe it's something else, or like this force,
like the eccentric contractions or something.
So it was a pretty good article that I read a couple of times.
That does make sense. That way.
Another question is, well, I guess we kind of answered this. Why do we see muscles grow better at longer lengths? Does the extra growth only come from sarcomeres added in series?
So I guess we sort of covered that. But I guess at the long length, like I said, you're sort of providing that like a maximal tension stimulus on your muscle, where you're both generating the active force, but also you've got a greater passive force stimulus. And then, it could seem to stimulate sarcomeres both in series and in parallel, based on some of the studies that are out there. So
and I think people need to understand too a little bit is like, I don't think a lot of the answers that if they're coming on this podcast to expect like a straightforward answer, like, okay, like this is it, I don't think they're going to be happy, because
it is the answer for a lot of it is like it'll depend on several factors. But yeah, I think in general, like a main conclusion across a lot of it is that like the eccentric phase of your resistance training is super important for putting tension on your muscle so that it'll grow, whether you're talking about in series or in parallel. So yeah, basically like don't rush through it. You want to be controlling that eccentric phase of any lift that you're doing if you want to maximize the growth stimulus. If that makes sense.
And
That's like
I
the overall conclusion I think that we can take from all of them.
Yeah, and I think something I think about is like, okay, well, let's say concentric contractions decrease. I think they decrease sarcomere in series.
Mm-hmm.
If like a muscle can experience passive tension or sufficient whatever it is that causes them to add series sarcomere numbers, if we decrease the sarcomeres in series, I guess now they can experience enough to add sarcomeres in series back to their resting length, maybe.
Yeah, for sure. That's yeah, that would for sure be the case. Yeah, like if it's lost sarcomeres in series and then you apply a stretch stimulus to it, then yeah, it would add sarcomeres back. Yeah, it would be able to do that. So
yeah. All right. So the next question was why are muscles more damaged when trained at longer lengths?
mm-hmm.
If you're familiar with that, because I know it's like a real complex thing.
Mm-hmm. Yeah. So the main thing really is that like at the longer length, the sarcomeres, like the smallest unit in your muscle, those are going to be stretched. And they are just more susceptible to stretching so much that they break, basically. So then the muscle becomes damaged, then it has to go through a process of repairing that damage or even growing so that it reduces the chance of damaging the muscle the next time it's exposed to that stimulus.
Yeah, I've seen a lot of people talk about also on channel where it's like, whoa, when you stretch a muscle, it brings in like calcium, and then that damages the muscle too. I've seen a lot of talk about that.
Possibly, yeah.
But I don't know. I mean, I don't know how much more damaging it is.
Mm-hmm.
I feel like people are like extrapolating something from a paper and then saying like, well, let's apply this to resistance training. And then, well, I mean, it adds up, but with the data, I haven't really seen a lot of the data. I don't know if you have about muscle damage at longer lengths.
Mm-hmm. Yeah, I don't know about like definitely very high intensity eccentric contractions do induce muscle damage. Like when you look at it, like I sort of mentioned earlier, but like transmission electron microscopy, like you can actually visualize the sarcomeres in a muscle biopsy, and yeah, you see that they're just like destroyed, like broken apart and like all wavy, and so yeah, like very high intensity eccentric contractions that will stretch those sarcomeres so much that they break apart and become damaged. But then the muscle, like it's not necessarily a bad thing unless you do it like all the time. That could be bad for you. But yeah, like it causes the muscle to go through a repair process that makes it sort of protected from that damage in the future.
Yeah, so they're like disrupted, right?
Yeah, exactly. Yeah.
Yeah. All right. So the next question after that was when the muscle is damaged, would it add, or when it's damaged during eccentric contraction, would it go first into adding serial sarcomeres or adding sarcomeres in parallel? I feel like that's a pretty bland question, but
so that's a huge question in the muscle damage literature that surprisingly nobody's really looked at. Like if one like damaging bout of eccentric exercise causes you to then add sarcomeres in series. We're actually doing a study in our lab right now where we're looking at that question. There's an undergrad in our lab that's leading that. So yeah, hopefully I will have an answer in like a year from now. But yeah, surprisingly, so the general thought is that sarcomeres are added in series to protect the muscle from damage from from the eccentric contraction because it would like reduce the tension associated with stretch. But yeah, I would think that you could also probably add sarcomeres in parallel when you go through that repair process. But yeah, the general theory on it is that sarcomeres are added in series after that eccentric induced damage.
More so than parallel, because
yeah, because you would be preventing the stress associated with that stretch in the future by adding sarcomeres.
Yeah, I mean, there's still a force generating component, but like you said, maybe like the greater stimulus was from that stretch causing the damage.
Exactly. Yeah. Yeah.
What else? And then two more questions. Last, this one being, does a shift in the length tension curve also increase the length of the length tension curve? Or is it just a shift keeping the same length?
That's a great question. So yeah, I think it does. So yeah, it is. You do shift like to the right, but I think generally, yeah, it does like widen the whole length tension curve as well. I sort of alluded to it earlier, but yeah, it'll basically increase the range over which your muscle is closer to optimal force. So yeah, it'll like widen the whole length tension curve as well, generally.
Yeah, 'cause I always thought of it like the same thing, and then it just shifts. But
there
I
are
think
some studies
sometimes
that look like it just shifts to the right. It might depend on like what level of the muscle you're looking at too. I think a lot of the ones that look at like the whole muscle tendon unit or like the whole joint do see that like the whole thing widens in addition to it shifting to the right. So it's kind of cool.
Yeah, so I guess like the optimal angle, I guess, can increase in length rather than just shifting through different part of the
so
curve.
yeah, it can shift to the right, but then also extend on top of that. Yeah.
Cool. And the last question is isometric exercise at long length or eccentric in a range of motion, mainly at short muscle lengths, which would be better for sarcomerogenesis?
Ooh, that's a great question.
It's from another researcher.
If I were to speculate, I would guess isometric training at a long length. Like if you're at like pretty like you want to be at a pretty maximally stretched out length, compared to just eccentric training like at a very fixed like short muscle length. I don't know if like I would guess that like it might just be too restricted to get much of the stretch stimulus out of it, compared to what you'd get when you're like doing a maximal isometric contraction with your muscle fully stretched out. Like you'll definitely feel that stretch and like the tension that's being generated there way more than doing eccentrics from like here to here. So yeah, that would be my guess. Someone would have to. That would be a cool thing to test out.
Yeah, I mean, I guess because like the muscle wouldn't really be that lengthened, but I mean, I don't know if it's like
compared to if you like are fully stretching out your muscle and like you're doing a maximal isometric contraction. I think that would be probably more. So that would be my guess.
Yeah, and I mean, with that being the final question, I guess we could finish off with: is there any like upcoming research that you're working on, or maybe things that you'd like to see in the future that can definitely help move forward this topic in the industry?
Yeah, so all the stuff that I've been working on for the last like year or so is in old rats. So yeah, I'm basically trying to determine. So first, I wanted to see if sarcomerogenesis is possible in old age, like in muscle, in like an older person, or I'm using rats, but there would have to be some kind of translational aspect. And it seems like it is, but from there, I went on to figuring out what would be like the optimal intensity for eccentric resistance training to cause an increase in serial sarcomere number in an aged muscle. Because muscle in older people and older animals is more susceptible to or has a harder time recovering from muscle damage induced by eccentric exercise. So you kind of have to be careful in how intense the eccentric contractions are. So yeah, right now I'm trying to figure out the best like training stimulus, I guess, with eccentric contractions for muscle in older rats, but again, could maybe be translated to humans, hopefully. So
cool. So, yeah, I'm sure a lot of people would also like to see like more on like the training at longer lengths too. Everyone's obsessed with that now.
Yes. Yeah, that might be
but,
the next next place that it ends up going after that.
But yeah, like you said, the stuff is really expensive. I wish somebody like Jeff Bezos was obsessed with hypertrophy
That's true.
and they could just
Yeah, could you
fund.
imagine the stuff that would get made? That'd be pretty cool.
Yeah, because a lot of it is just expensive stuff where it's like
Exactly. Yeah.
80,000 machinery
Yeah. All the
kind of
equipment.
thing.
Yeah. Sure.
But yeah, I guess we could finish it here. Before we end, Avery, where can people find you and I guess give you a follow or ask
Yeah,
questions?
so you can follow me on Twitter at Hinks Ave. I use that pretty regularly, or on LinkedIn if you want to see. I post more like I post basically the same stuff on Twitter that I post on LinkedIn, but I guess I get a little more in depth on LinkedIn with the stuff that I do, just because there's no character limit. If that makes sense, but yeah,
Yeah.
either of those people should be able to find me.
All right, cool. Well,
Yeah,
thank you guys for watching, and I will see you guys in the next episode. Thank you, Avery, again for
yeah.
joining us in this
Thanks for having me
podcast.
on. It was a great time.